Hello everyone! This is the homemade experiment of DNA extraction in strawberries of the group 1B. My name is Paulo Ortarte and I'll talk to you a little bit about it. All living beings have genes, which are genetic information storage units that have segments of DNA that contains information of all body cells. Strawberries are the fruit of which is gotten the most DNA and are also octuploid, which means that they have 8 identical sets of chromosomes. These circumstances make strawberries' DNA easy to extract and see. Our experiment consists of 7 steps, and besides strawberries, it requires the following material and reactives. We are going to need a Ziploc bag, and stainer, and small spoon, some tweezers, a syringe, salt, water, beakers, and ethanol. As a first step, we're going to measure 15 milliliters of ethanol, of which I already did, and we're going to put this into the freezer.
For the second step of our experiment, we're going to need a glass. We're going to need 90 milliliters of water, 10 milliliters of dish soap, and a teaspoon of salt. We're going to mix all of this, and we use dish soap because it makes it possible to break the plasma membranes and cell walls so that it can be able to access the cell and enrich the genetic material. And we use salt because the salt breaks the protein chains that bind the nucleic acids in order to release the DNA chains.
Now we mix the dishwasher, salt, and water. And this will be our DNA extraction solution.
Hi teacher, continue step number 4. And we put a strawberry in a zipper bag. Okay, we put the prepared solution. Okay, and we expel all the air contained. Okay, thank you.
What we are going to do now is that we're going to put our strawberries in the bag. I already put mine in the bag, and you're going to grab your mortar or your plastic glass and you're going to smash them very, very well until we have a consistency like jelly. Once we have smashed it, it should look like this. This is one of the most important steps, so make sure you do it with patience until you get this type of consistency. I'm going to explain why this is so important. As we know, vegetable cells are covered by a wall of lipids and proteins. By breaking this wall that protects the cell, we can get to have a more clear look of the DNA. In further steps, we will be adding another substances that will also help us to see the DNA of the strawberry, but this one is one of the most important. So make sure you're patient and you do it very well.
Hi, my name is Luis. For the next and one of the last steps of the DNA extraction of the strawberries, we will use a beaker, a small spoon, the strainer, and the strawberries that we had inside in the bag. Throw the strainer, put into a beaker, pour the content into a beaker. Use the spoon to press the strawberry pieces against the strainer. We will do this slowly so that our samples do not spill.
Finally, add 50 milliliters of ethanol and observe if there is any change in the mixture. Why use alcohol in this experiment? Because alcohol attracts water and makes easier for the sodium interact with the PO3 and precipitate DNA. So it's more efficient to use ethanol or isopropanol. When the molecules are insoluble, they clump together and they become visible. The alcohol separates the DNA for other cellular components which are left in the equation solution. So can you see this part is DNA for strawberries. Conclusion: strawberries are ideal for this experiment because they are the fruit with the highest about DNA of them. Can you see that? So, stretch DNA for strawberries by cell lysis by a homemade method giving us an overview how to obtain the genetic material of the fruit in the macroscopic way. So this is it. Thank you for